Let's go to the class. So let's see. So we're in day 2. Yeah, so it was this one, right?
Yes, yeah.
Okay, so what I would do is I would just use the notes because they come right from the textbook. So they're probably pretty much in sequence, I would bet. Let me just take a look. It's probability, segregation. Yeah. Were there ones that you had a question on? Anyone in particular?
Um,
Pull
Not.
that.
I'm only on question 5, but I haven't answered that one yet. But I
The
think,
actual question 5 that's on here.
yeah, that's where I've left. That's where I left off.
Okay. All right. Because on the Edge Elastic, they're not necessarily matched up because I don't ask every single one. So how do geneticists use Punnett squares? It's all about predictions. It's all probability. Basically, you're trying to predict what the probability is that the offspring will have a particular genetic sequence. And when you're talking that, you have to either specify genotype or phenotype. And the reason I say that is because genotype is the actual letters. I don't have this open on my pen thing. I can though. But right here where the Punnett square is, do you see my cursor moving around?
Yeah.
Okay, so the letters themselves, that's the genotype. The phenotype is what the physical characteristic is. So a geneticist might use the Punnett squares to predict a particular genotype and not necessarily the phenotype. And I'll give you an example. If you wanted to know what your chances were of being a carrier, so in other words, both parents, your parents, have a genetic disorder that they don't express because they're both carriers. But you might be interested in marrying somebody who does have this particular disorder, and you want to know if your children will have it. Well, that depends whether or not you're a carrier. See what I mean? Because if you're not a carrier and all you have are the capital letters, then the best that can happen is your child will be a carrier. At least they won't have the disease, but they'll be a carrier of the disease. So what they need to know is if both of your parents are carriers, you need to know whether or not you're a carrier, because then that would change the probability of the child having the disease. Does that make sense? Or is that's a lot, right? I'll show you what I mean. I'll show you what I mean. So let's just do a Punnett square. Go back out really quick. So here's our Punnett square. And dad, your mom, this is what they have. Little t is the disease, okay? And when they had children, these were the possibilities that they could have.
Oh, but there's
All right,
a
this
great.
is the disease child. This is the kid with no disease, and this would be you. You're a carrier. So now this is you, and you happen to be interested in marrying someone who has this. Okay, now it's a disease, but still, you want to know whether it's life-threatening. Obviously, it's not life-threatening, or your partner would not have achieved adulthood. But it may be some disease. Okay, it could be something that's like medically requires a lot of medication, which would be a very expensive person for health insurance reasons. So you want to know what are the possibilities your children will have it. Well, it depends whether or not you are this or you are this. You see what I mean? We know you're not this because you would have the disease. So you're either one of the three, but we don't know if you got the carrier or you didn't. And so they would do a geneticist would do a genetic, like they call it like a pedigree, and they would be able to figure out whether or not you were this one or this one, because that depends on whether or not you're going to have a child with it. Because if your husband to be is this and you're this, well, then it doesn't matter. At least you know your child's a carrier, but at least you know they're not going to have it. But if you were this, well, now you have a pretty good chance. You see what I mean?
Mm-hmm.
Because
Yeah.
this would be this, and then you would have there'd be a 50-50 chance that they would have it. And if they didn't have it, they would definitely be a carrier of it. So that's like the long the long answer to that, but that's what a geneticist would use this stuff for. It's mostly when you're ready to have children, they'll check the life history and they'll do stuff like that. Trying to think. You'll definitely have questions for 7, 8, 9, and 10. So you want to make sure that when we're talking genotype, we're talking about the actual letters, the physical letters, a capital T and a lowercase t. That's a genotype. If the two letters are identical, then we call that homozygous. If the two letters are one's capital and one's lowercase, then that means you have a variation of the trait. You got one variation from mom, one from dad, but the capital letter seems to overpower the lowercase letter. When there are two different letter cases, that would be heterozygous. And phenotype is when you're talking about what the letters code for. Like capital T, lowercase t, does that mean you're tall or does that mean you're short? So what they actually code for is the phenotype. I'm just trying to go through a couple others. Question 13 will probably be the hardest one because you'll have to probably draw out the choices. So like, let me see. So this is section 11 too. I should be able to open this up. 1, 2. All right. So for 13, you're going to have to, it says in an F1 cross between two hybrid tall pea plants, half the F2 plants will have two alleles for tallness, capital T, capital T. All right, that's a lot. So let's just make this red so it's easier to see. And we can draw it over here. All right, so in an F1 cross between two hybrid tall pea plants. So you are crossing two plants that are this. So you basically have this. Oops. And this. That's what you have. And so you're going to have a capital T, capital T, capital lowercase, capital lowercase, lowercase, lowercase. Half of the F2 plants will have two alleles for tallness, capital T, capital T. Well, I don't see anything here that has two capital T's except for the first one. So half of the F2 plants. That's not true. So you know it can't be A. I'm just pointing out how you go through each of those choices. Do you see why it's not A?
I see why it's not the capital T, but
Why? Okay, so in F1, so this is our cross, capital T lowercase, that's what we're crossing. And that's just those are what we call hybrids. So they're heterozygous, and they're telling you what they are. So there's a lot of information in this first sentence from here to here that just repeats itself. And they're making a statement that half of your 4 boxes will be capital T, capital T. That's what the rest of this says. Half of the results will have two alleles for capital T, capital T. They'll have two common alleles. And what you is no. Only that one there is capital T, capital T. The others are capital lowercase or lowercase, lowercase. So only 1/4 of the plants or the next generation had capital T, not 1/2.
okay.
Do you know what F1, F2, what that really means?
I did it yesterday. I wrote down all the answers, but
All right, it's here.
Isn't like F1 the beginning stage?
P1.
The first generation.
That would be your P1 generation.
Oh, okay.
And then you get your F1, and then you get your F2. So generally what happens is your P1 generation is when you cross your purebred with your purebred. So these are both homozygous. All right? You follow? Okay. So here's a purebred tall. That means it doesn't have the gene for short. And this is a purebred short. It doesn't have the gene for tall. But all of their babies all of them, no matter what you do, are going to be capital T, lowercase t. So what happened was Gregor Mendel, when he took all of his, so here's his tall plants, and these are his short plants. So Gregor Mendel spent all this time taking all of his tall pea plants, and he cut off all of the reproductive parts of the plants. And he basically self-pollinated the tall plants. So he purposely made sure that after generation, let's say you did this in a greenhouse for 6 weeks, and let's just say pea plants grow really fast, like every week. So you have 6 generations, and all you got were tall plants. You would assume that any of the seeds from any of those plants would have been tall. You would have weeded out the recessive trait. Does that make sense in what he was trying to do?
Yeah. So
okay.
Yeah.
Okay. So then he does the same with his short plants. So after time, after several months, he's essentially got within confidence a seed that is pure for tall and a seed that is pure for short. And when he put the seed together, so now let's say the plant. So he grows those plants. So now he takes the female part of one plant and the male part of the short plant, and he pollinates them. So now you're crossing a short plant with a tall plant. So he wanted to know what would my result be. And all of his plants were tall. So the first thing that would come to your mind is, well, what the heck happened to the short? Right? I mean, logically, what happened to short? It disappeared? Yeah. Did the tall eat the short? Like, what happened? He doesn't know. It was 1800 something. He has no idea. So now he goes ahead and he takes all of his short plants. Remember now, he did not know that this is what the letters look like. All we know is that we now know that the short was being hidden, but he didn't know that. So he took what we call his F1 generation. That means he took his pure tall, his pure short, and he got a plant. Then he bred those plants together. He inbred his F1 generation. And what happened was when he did this, and you do this Punnett square, right? I should have made it a little bigger, but that's okay. So when you take this, that's what one of these are, and you cross it with another one. Let's just make sure we get this right. So now, all of a sudden, in this generation, he gets some of these. He's like, all right, well, the other generation was tall. He gets some of these. Remember, unbeknownst to him, he didn't know that they were carriers. He's like, okay, it's still tall. And then all of a sudden, he gets some of these. And he realizes, well, how's that possible? Because the short was in my parent generation. It skipped this generation and then came back out in this generation. See what I mean?
Yeah.
So all of a sudden, Gregor Mendel starts to have this idea that genes must segregate, that they must be made up of 2 parts, and that they separate from one another so that one version doesn't always go with the offspring. That's what the law of segregation is.
Okay.
So when you see law of segregation, that's what they're talking about in here. This is law of segregation. So if you look at, say, choice D, segregation occurs according to Mendel's model. Absolutely. Because if you look at Mendel's model, you'll see that the 2 purebreds stayed. The resulting generation of those 2 purebreds, the recessive wasn't shown. It's there, but it's not being expressed because it's masked by the tall, and then it pops up again. And so that shows segregation. Otherwise, there's no way to explain how the short gene would skip a generation.
Okay. It does make more sense looking at it like as a picture.
Yeah. And then probabilities, predict averages. So probabilities don't really make predictions, just so you know. It's like flipping a coin. If I flip a coin 10,000 times, what's the chances I'm going to get heads on the 10,001 flip? still 50%. It doesn't matter how many times I flip it. It's a 50% chance. Like someone saying, what are your chances of having a boy or girl? 50%. Well, you had 10 kids. They're all boys. What are your chances of having a girl? 50%. It's 50% every time.
Yeah.
So there's no way to make a prediction. So that's what this is. The 15 through 18 are questions to just make sure that you're not guided towards thinking that probability is a predictable. It gives you some estimation of what could possibly happen. And that's like, so then what I did was I gave the kids same idea as last week. I opened up Wednesdays because I didn't know who wanted to work a little bit faster than others. And there are some practice problems. I only have you doing the first 6. So even though there are problems, you will have the others to do for the like maybe Thursday. So if you wanted to try the others, you can. But I only had the kids do the first 6 because there are a couple that they can't do on the second 6. And there's one that I'm blood typing. I think that's a little confusing. So the hardest one on here is number 6. Actually, 5 and 6. Okay. 5 and 6 talks about what's called a test cross. So it's just about using your creative thinking about how would I, remember what we just did. Remember we were saying, oh, I deleted already. But remember, we don't know, like, for example, if someone's a carrier, we don't know, like, if you're heterozygous for a trait or if you're homozygous for a trait. In other words, if you know that there's a family history of an illness and you know it's a recessive trait and you don't have it, well, we don't know if you are capital and that's why you don't have it, or if you're capital lowercase and you're a carrier, but just because you have the dominant allele, you're not showing it. Well, there is a way to go backwards and figure out which one you are. This is not something you would do with people because you don't do test crosses with people because that would be like having a child to see what your genes were. Like that's ethically, there's so many things wrong with that. But this would be like for plants. You know, how could I go back and figure out if the plant was a carrier for a trait? There is a way to test another known plant, and based on what you get as offspring, you can figure that out. So it's a Punnett square problem. You're basically looking at the Punnett square and you're trying to figure out, what could I test it against to see? It's just a critical thinking problem. So most of these are percentages. That's what most of these answers are. So you're just going to have to do the Punnett square and then go ahead and just do, the percentages. If it's 1 out of 4, it's 25%. And so forth. So I made this in Edulastic, and that's it. They'll just be, from here, we'll do incomplete and codominance, which is this, nothing is going to change this week in Punnett squares. So if you feel like you're comfortable with the Punnett squares, that's as far as we're going to go. The only thing we're going to do is we're just going to introduce a couple twists to the vocabulary, and they are things like this. And don't panic. We're not talking crazy stuff here. So, for example, up until now, we said capital T is tall and lowercase t is short. And this is what we call the dominant.
And recessive.
Yeah, and this is the recessive.
Trying to memorize it.
Right. So there are, and Gregor Mendel would not have known this, but as we started to learn more about genetics, it turned out that there are a lot of, for number one, there are, it's not clear cut. In other words, it's not always one combination of letters codes for a trait in your body. Don't get me wrong. There are some that are as simple as like one letter combination. But like in the case of your eye color is actually a combination of 2 or 3 different genes that are talking to one another. And the way they talk to one another also depends on what the combinations are. So it's a little bit more complicated. But one of the things Gregor Mendel did not know is that some genes are blended together. So let's say, for example, we said R is red and little r is white. based on what you've learned, you would say that if I had a capital R, little r, that it would be red. There are instances where a capital R, little r would actually be pink. And this is called incomplete dominance. You want to make sure you record that. Dominance. And so that means that there's a blend. And you'd have to be told that. You wouldn't know that. So I don't want you going around memorizing, oh, when it's capital R, little r, it's not red, it's pink. No, you would have to be told there are two traits. Red is dominant over white, or no, red is incompletely dominant over white. And one parent is capital R, and the other parent is little r, what would all their offspring be? Well, they would all be pink because they'd all be capital R, little r. But you would have to have been told that they express incomplete dominance. Make sense?
Yes.
Okay, there's one more. The other one is, right, you could, we'll use one that just makes a little more sense. So we'll say capital B is, we'll do it over here. So we'll say capital B is black and little b is white. If you had capital B, lowercase b, obviously that would normally be black. Oh, right. But in this instance, and you'd have to be told, you could be polka dotted. So like a Dalmatian. And this is called co-dominance. That's another word you'd have to be told. So you would have to be told in the question, the parent is co-dominant, or is homozygous co-dominant for black, and the other parent is heterozygous for white. So that means one parent would be homozygous co-dominant, they'd be this, and they would be with a heterozygous, what are the chances that their offspring will be polka dotted? Okay, so you would be making a Punnett square, and you would have your capital B for this one, and you'd have your capital B lowercase, capital lowercase, and so 50% would be polka dot because they would have the two, and we'd be told that they represent co-dominance. So all I'm saying is that on Thursday and Friday, in addition to regular dominance, the Punnett square and the process and everything will stay just as it was when we ended Friday, and we've been doing the beginning of this week. The only difference is you just have to be on the lookout if somebody says that the level of dominance is either A, incomplete dominance, in which case the heterozygous or the capital lowercase would be a blend of the two traits, or if the question said co-dominant, then the uppercase, lowercase combination would be a polka dot. They both express their color. If it was the red and white, it would be a polka dotted flower, like a carnation.
Okay.
Other than that, there's nothing new. When Monday, probably not Monday just because it's the weekend, but probably, you know what I'll do? I'll probably have like an end of unit assessment for like Tuesday, but I'll open it up on Monday if the kids want to do it. But if they don't want to do it, then they can do it for Tuesday. And then they wanted us to have like an opportunity for kids to like check in and tell us a little bit about what they thought about distance learning in case we start this way. You've been on so much, you don't worry about it. You don't have to participate unless you really feel strongly about voicing your opinion about stuff that can be done different. But you're going to expect the same thing in all of your classes. So just if you're wondering what week 11 looks like, that's what it should look like in all of your classes.
All right. So
it's mostly to make up work.
All right. Yeah, Mr. Dalton said it'll be like surveys stuff for next week, so boring stuff.
Yeah, I mean, you've participated every day, so you probably have made up for quite a few students who haven't. So I thank you very much for that. But that's what's going to happen for next week. And then, like I said, this week it's just a continuation of these Punnett squares. You'll have the problems for tomorrow if you want to try working on some of them. They're 1 through 6. And then if you want to try the ones on the back, you can. The ones that are on the back, do I have them in here? I don't know if I have my handout with me. I do. If you want to try them, don't do number 9. Number 9 you can't do. It's a blood typing one. And I think you can do all the rest. So
Those ones are due for tomorrow, but they're posted.
the ones that are due for tomorrow are the 1 through 6. And then what I'll probably do is on Thursday, I'll have another live session Thursday, probably go over 1 through 6. And what I'll assign for Thursday is probably 7 through 13, but you won't do number 9. So it'll be another 6 of them. And then Friday, I'll go over those. I won't introduce anything other than review what I just told you about the incomplete and codominance and just show kids what would happen if you had a heterozygous, how it would be blended or be polka dotted. And then that would be it. We would just end on Friday with that. And then on Monday or Tuesday, I'll post an Edulastic. And what I'll do is I will post it'll basically be the quiz that you guys took last Friday with like maybe 4 or 5 Punnett square problems on there.
Okay.
And I'm pretty sure that and just double check when you get a chance, make sure that you can see your responses to that Edulastic because it's of no value to me if you can't review the ones you got wrong.
The one we did, that one?
Yeah, the one you just took on Friday with had all the different graphs. You and I went over the graphs, I think, the day before the actual quiz.
Yeah, I did.
Yep. So it'll be the same questions. You know, the things might be a little bit different, like the same graph, but I might change instead of like, for example, the one that asked about the beta cells and the gamma cells and why which one was in the synthesis stage. I might change that question to something a little bit different, but use the same graph. Same skills, just a different stage. That's all.